This is not for each of us whose family are affected. They are not just another number. We're losing our loved ones, our mothers, our fathers, our wives and husbands. And it's impacting on every one of our lives. And therefore, it is momentous. It is necessary. Now is the time when we need to act to address these problems. This problem, the problem of cardiovascular disease in particular, cardiometabolic diseases, including diabetes, obesity, are a major problem for CARPHA community. This is a report which was published in the Lancet Regional Health Americas in 2025. And as you can see, it highlights the incidence of cardiovascular diseases globally. And it has been recognized that the CARPHA region and specific countries, including Guyana, are right there at the top. Not in the right way, in the wrong way, because we are having much worse incidence. We're looking at the comparative burden of cardiovascular diseases and deaths resulting from these. And we can see in the light blue and the orange that the Caribbean community and Guyana in particular are actually showing very high statistics. Those are not the statistics we want to be high at. Those are the statistics it is incumbent on us to start addressing. And in addressing that, as a scientist, I'm a molecular biologist. For many years, we've been looking at the mechanisms driving these diseases. Most of us will recognize the problem before us. But let us think for a moment. We have our families or ourselves, we have chest pain, or sometimes we get a little bit breathless and we put it down to just a bit of tiredness or whatever. When do we go and see our clinicians or doctors? We go to see our clinicians when the disease has caused irreversible damage within our cells, within our tissues, within the heart. Irreversible damage that cannot be repaired. And as such, I will suggest that our current care and treatment of such diseases is reactive rather than proactive. We are treating the symptoms of the disease caused by damage, often irreversible damage to the tissues. And that is all that we can treat. Therapy, in this case, we start that at the end of the disease process. And we will have limited outcomes from that. But we are being reactive. We're treating consequences, not the causes. And that there highlights for me what the significant problem facing us within the scientific community, within the public health community. There is a pre-symptomatic clinical blind spot that we must address. That is the time when the damage is happening, when signals are being sent out because the heart is starting to be very stressed, but we are not able to detect it in sufficient time to do something about it. So my talk today will focus very much on that clinical blind spot as a challenge. In this case, cardiovascular disease, but it relates to chronic diseases in general. Let's look at the heart in particular, the heart and blood vessels. We can see that the irreversible damage doesn't just occur in the heart. It affects the blood vessels. It is influenced by the fat cells in our body, the adipose tissue. It is influenced by inflammatory changes. And all of that occurs over a long period of time. It occurs over decades. None of this is overnight. And in the first couple of decades of sort of the symptoms arising, of the changes arising, there are no symptoms. We cannot detect it. And by the time we have symptoms arising, we will see, for example, in our blood vessels, it's already clogged up. Atherosclerotic plaques have taken hold, and there is very little we can do about it. The heart is damaged. The cells cannot be replaced because these are highly specialized cells. So what are we going to do about it? We treat the pain, we treat the symptoms. And what we cannot address are the starting point of when this damage is actually happening. So that is the challenge we face. That is the challenge we all as a community need to be able to understand a little bit better. And in the many years of studying this, what we have found is, understandably, it presents a real challenge to us. Because if we think about the heart, it's an organ that sort of has to work constantly. It cannot take a rest. In a normal heart, everything's going along quite nicely, working. But if you have stress, if there is increased demand on the heart for any prolonged period of time, adaptive changes start to take place. They will happen in the cardiomyocytes, the very specialized heart cells. It will happen in the blood vessels. It will happen in the supporting cells in the heart. If we do not recognize those changes, that's when the disease process starts. And when we start getting maladaptive changes, when we start getting damage that are becoming evident as symptoms, by then often the damage is irreversible. We now currently, our treatments are sort of somewhere between phase 2 in that diagram and phase 3, when the disease is starting, but it's remaining silent, and the maladaptive changes are taking place. By the time we get to irreversible damage, there is very little we can do apart from providing support and hoping for the best. So we have, if we look at the mechanisms that people have been working on for many, many decades, we have identified a window of opportunity, a window where research is absolutely essential to be able to help us to understand, to illuminate those questions that are arising, so that when we can understand what is going on, we can then try and improve outcomes. So that is the challenge that we have to face. We need to now shine the light of research on understanding that adaptive phase, during which there are no symptoms that are now evident, but before the maladaptive changes start giving rise to symptoms. Now, those of us who are actually, all of us will have some form of basic molecular biological background. And we know that a lot of that arises at the molecular level. It's at the DNA, the level of the DNA. All cells have the same DNA, but something that must translate from that to generating the specialized cells, to generating the responses from when the heart is sort of working normally to when it's stressed. Those are the things we need to understand, and that's at the genetic level. That's where we need to now look at. And what I would like to just use an example to show is how asking the right questions can be used to reveal the mechanism and then create the solution. Now, all of us will be familiar with Ozempic, the GLP-1 agonist. that has revolutionized the treatment of obesity and diabetes. But if we think about it, we see this as groundbreaking and we think how wonderful. But that discovery has taken decades of scientists working across the globe to understand the mechanism. And it wasn't until we were able to understand that this peptide produced in the gut, influenced by the gut microbiome, the bacteria living in our gut, until we could understand that though the peptide produced in our gut can influence the adipose tissue, the fat cells, to produce adiponectin, can signal to the brain, particular regions of the brain, to suppress appetite, can trigger changes in the pancreas to stimulate insulin to cause glucose uptake. Until we could understand these connections and these mechanisms, it was not possible to harness that. It's only after we understood the mechanism, we could actually then understand how we can harness that to address such a major problem facing the world today. So asking the right question is what we need to be doing right now. But there are two problems amongst many others, but two I'll highlight. How do we find the solution in that window that is currently invisible to us? It happens at the molecular level. It happens at the level of our DNA and our RNA and our protein. We cannot experiment on humans for obvious reasons, but we need to be able to test the changes that are taking place at those very early stages in the heart, in the brain, in these different tissues. And the other thing is to be able to look at how these different changes are occurring and what controls them. So in addressing those issues, we've had to look at innovative solutions to this. What do? We choose in vivo models. We take mouse models where we can use technology that's available to us to change a single gene, to then be able to use that to test how and what we can do about it. So we can use cell lines, we can use other models. And through that, I just want to show you how we might be able to link such models to reveal answers to questions. So for example, we have been working on a transcription factor, master regulator that switches genes on and off. We have shown that in generating one of these, we can measure the heart and we can show that the male hearts have much worse outcome when compared with female hearts. You could see on this measure of the contractile dysfunction there, we can see the female hearts, they look very similar in terms of the contractile function. The male hearts, you could see that the red bars are actually much lower down. It seems, therefore, that loss of this transcription factor can cause poor heart function. Similarly, we can take the hearts, we can section them, we can do analysis of them. And the blue stain here shows you where you might have specific types of tissue, scar tissues. You could see in the female hearts, we have no abnormality when the heart is stressed. Look at the male hearts. You could see the blue spots, the blue marks show where you have scarring around the blood vessels. In the ventricles as well, we get profound scarring, scar formation, which means that the heart is unable to contract effectively. We can use these animals and test them on how they respond to exercise. We show that the females mutants as well as the controls can exercise as well as each other. And their heart improves with exercise. But look at what happens with the males. The red line that you could see at the bottom there is the mutant mouse, which lacks this protein. And you could see that the males there perform very, very poorly. And if you look at their heart function, we can see that the ejection fraction, the output from the heart on every beat is actually significantly reduced. So all of that is just showing us that there are profound differences in how the heart behaves when this protein is there or not there. And we have been able to also test using microscopy, electron microscopy, so we can look at the detailed structure of the muscles to show that we have got disruption of the connection linking the two heart cells together, the intercalated disc, which is absolutely essential for the heart to be able to beat as a single unit. So any conduction of electricity across the heart is essential, and any disruption of that will create abnormal signals, will cause the changes we see in ECG. And sure enough, when we look at that high magnification in the mutant male hearts, that's exactly what we see. We see that that structure is completely disrupted. So these are just some evidence. I don't want to bore you too much with details of it, but it's just to show that these types of asking the question, using the resources we have, can answer questions to show how we might be able to map what is going on within that window, clinically silent window. And what we have gone forward to do is we've mapped the whole genetic platform across the male and the female hearts. We took the mutants and we took the wild types. And what we have shown there is that the males have very, very different profiles. They separate like two distinct sets of behavior. The females, on the other hand, they don't show that much difference. And when we analyze that, we could see that at the genetic level, that in the males, we got 96 genes which are reduced in the knockouts, and 190 which goes up when this master regulator is knocked out. You look at the females, we have 9 genes in all that's changed. That in itself gives us an insight into the window where silent changes are now taking place, which we cannot see. But that allows us to understand the disease process a little bit more in that sort of silent phase. We can look more closely and we analyze those, and we see structural changes. We see contractile changes in the muscle and the structure of the proteins themselves, of the heart itself. We see energy production is disturbed. And all of this, we put it into a sort of bioinformatic analysis, and we could see how many human diseases are linked when those genes are modified. So that tells us directly that knocking out this particular gene, this master regulator, can cause these human diseases linked to electrical dysfunction, linked to contractile dysfunction, linked to cardiomyopathies, heart failure. So this is now telling us 2 things. One is that BRAIN3B, this transcription master regulator, is critical for controlling heart function in male hearts, but also that if it is lost, it is directly associated with heart disease in humans. The second main question that comes up is, why is it that the females do not have the same adverse effect when this is lost? Now, that maps out to another very interesting sort of known outcome, that estrogen protects females. Young, healthy, premenopausal females are indeed protected against heart disease. But if we look at that, any of the factors that change, like obesity, like diabetes, can prevent those sort of protective mechanisms. And what we know is that this BRAIN3B transcription factor, this master regulator, interacts with the estrogen receptor and modifies the behavior, the fate of cardiac cells. And so now we're trying to understand, which we can show here, and again, I'm not going to go into details, but we know that in the female knockout hearts, you get an increase in this estrogen receptor, which seem to compensate. So that tells us, and again, two things. One is that estrogen receptor is protecting the female hearts because it can go up, but that the BRAIN3B transcription factor is absolutely essential to protect male hearts. Now, going forward, how are we going to harness these? What have we found? We know that silent onset of heart disease is linked to the molecular level, the changes that are going on in that window that we cannot yet see. We have identified a master regulator, which is necessary to protect male hearts and acts in the same pathway as estrogen receptor and estrogen in protecting female hearts. So we have mapped the factor that is necessary to protect male hearts. We also know from our study that we can detect BRAIN3B in blood from patients with angina. And we can show that that lowering of BRAIN3B is linked to the disease. So now we have a window in which we can actually perhaps provide a biomarker to look at early changes that might be a risk, provide a risk marker for disease, particularly in males. Or it might also apply to women where you have either postmenopausally or with other risk factors that increase the risk similarly or even more than in men. Now we have understood the mechanisms, we can ask, how are we going to apply this in a clinical setting? Might this have some application that we might be able to establish either for early diagnostics, for providing sex-specific care, and to target prevention of heart disease and the damage that we see at that late stage. So that is just a window of how we might be able to go from understanding, asking the right questions, understanding what is going on, and then being able to apply it in a clinic. So I'm hoping that with that, it might actually help us. Those are the sort of questions we need to ask within our own communities. How is it relevant? What are the things that are necessary to address within our own communities? Develop our infrastructure for these types of basic research, answering these basic research questions, because that is when, and it is the only time, when we will be able to stop losing our loved ones to these types of chronic diseases, so that we might be able to sort of move from what 30 years ago we had very poor resources. We had no laboratory facilities, a poor infrastructure, limited trained personnel, organization and drive wasn't there. Now I've visited some of the laboratories, I've visited the new hospitals here in Guyana, and I can see that there is the vision, the resources are there. I hear that in the talks that have been given. We have the organization leadership. We can see that in organizations like CARPHA, like the Ministry of Health, the CARICOM, PAHO, and WHO, in the activities that they are helping to pioneer. The infrastructure is there. We have more trained personnel. We're having talented communities of medical staff, our doctors, our nurses, our technical staff. We now need to be able to, as a community, drive that forward, take that to the next step, ask the right question. Let's build capacity. Let us then drive forward the preclinical research that would help us to stop the disease before it gets to being irreversible. So while at the moment we are stuck with a reactive care, let us move towards a tomorrow where we have proactive medicine. And that is the only way that we will be able to get to an outcome of healthier communities. And that is what I would leave with you here as a CARPHA community. Let us try and embrace the vision to inform politics and to act early. In going forward, I think I would just like to make a couple of acknowledgements. Firstly, the Ministry of Health in Guyana, particularly the Health Minister, Dr. Frank Anthony, and the Director General, Dr. Vishwa Mahadev, who have shown such strong commitment, the vision, and the ability to take things forward. And long may that last. I would like to see our younger generations coming along and show this sort of same drive and commitment going forward. And as I am here in Guyana presenting this, it would not have been possible without the inspirational Guyanese visionaries who have shaped and carved my outlook, Budra Mahadev and Rajkumari Mahadev. And with that, I say thank you very much. Thank you. I'm happy to take questions.
Thank you for an excellent presentation, Dr. Mahadev. At this point, any questions or interventions are welcome from the research community.
Yes, certainly. Thank you very much, Professor Vishwanti, for that very inspiring lecture. I'm happy to hear the community of molecular medicine, which is speaking at CARPHA. One of the things that you emphasize in is early detection. Can you comment on, I see one of your slides you spoke about low birth weight. Can you comment on the effect of low birth weight? What as an important marker in cardiovascular disorders? And you also emphasize the importance of that preclinical phase, that preclinical phase of cardiovascular disorders. Can you comment on that? And I think it would be useful if there are any pediatricians here. I think they should find your comments quite useful, because there must be a paradigm shift in the way we detect cardiovascular disorders. Thank you very much.
Thank you. That is an excellent question. But I think there are a few things that I completely agree that there is a need for a paradigm shift in our approaches, but it has to start from the understanding. A lot of the markers that are currently out are often linked to measurements of irreversible changes that have already taken place. So, for example, the damage from the muscles that actually gives rise to the current markers that are being used. At the moment, in terms of that very early window, there is still quite a lot that has to be done. And there isn't anything that we can use. There is obviously, you have the genetic component, which falls under a completely different remit. But in terms of the chronic cardiovascular diseases that are linked to the progressive changes that take place over many decades, that is something that there are still quite a lot of outstanding questions. There are markers that are available right now that the troponins and the sort of, looking at those types of clinical markers that are available, those will largely be within the window of you're having symptoms due to these changes, and these are then detected to say, yeah, there is this extent of damage to the heart muscles that are now being detected. However, I don't think that those still necessarily address the earlier window during which time there is an opportunity to be able to at least halt the damage so it doesn't get to being irreversible. And I think that is where I think the challenge still remains. And I think there are lots of people working at the moment to try and identify those types of markers.
Thank you very much for this presentation, Professor Vishwani. It's really nice. Just echoing Professor Cummings' comment about how wonderful it is to see basic sciences being integrated into the CARPHA program. We tend to move away from basic sciences bench research into more public health and more retrospective analysis. Focusing on clinical outcomes. But so it's good to go back to the basic sciences to understand disease mechanisms, and you provided a really clever example of how we can link basic research to clinical outcomes. And you mentioned troponin, and I was wondering, why can't we use something like troponin to assess. At the moment, troponin is used as a diagnostic tool for myocardial infarction. But you're saying that this is something that is progressive. So is there the possibility of using something like troponin, maybe I or T, to as a screening tool for as an early predictor of EMI?
Again, I think there have been various other markers that people are actually working on. The problem with the troponins and that is that they're released once there has already been damage to the tissue. Once the cardiomyocytes there are sufficient disruption to allow these factors to be found in the blood. Until then, we will not be able to detect them at anything that is meaningful. And again, it comes back to this question about, at that stage, with addressing something like highly specialized cells like the cardiomyocytes, which they don't have much of a sort of a proliferative capacity because of their specialization. So I think this is where we're stuck at the moment, that there is that anything that we can detect is based on damage that's already happened. So we're talking about, in that diagram, the phase 3, between phase 2 and phase 3, when we already have the silent going on to the symptomatic damage taking place. What you have mentioned, and I think I would love to see that being embraced a bit more, is for us within, like, I know Guyana, the Ministry of Health is talking about actively setting up sort of bench science to address these types of issues. We've got the capacity, we've got bright young doctors, nurses, technical staff coming along, training them, getting them to sort of be doing the bench science that can actually allow us to address the issues within our own populations here, which are unique. They are not going to be the same as what's in America or in sort of the UK or elsewhere. But we need to sort of develop that capacity within our Caribbean states. And so that is what, as a scientist, I could see the potential, and that's where I would be very passionate to see a lot more of that being embraced. I'm not sure if it addresses your question, but I'm happy to take this offline. But certainly it's something that at the moment is a limitation for clinical diagnostics and subsequent treatment.
Thank you, Dr. Mahdi.
Thank you very much. Sorry.
Thank you. Piedad Huerta Pajo, WHO. I would like to thank you for such a wonderful presentation and Also because we're speaking about a research focus on one of a huge problem, public health problem, one of the main causes of mortality and discapacity in the world, especially in the Caribbean. So thank you for that. Also, I take your last point because I'm so happy to see so many young people, researchers in this room, because it's important to see the value of making the correct question and also on strengthening the wonderful opportunity to focus research into primary healthcare, because I can extract immediately from your presentation many practical things that can go directly into primary healthcare, but also into how to do better research and have subsequent questions from your research to continue doing so. And just one more thing, because I was doing a little research around, and this is the first time in the conference that I see so many people connected with you in your presentation and not on our phones. So congratulations for that. This is not
Thank you.
scientific, it's just observation. Thank you.
Thank you very much.
Okay, thank you, Professor Mario. In interest of time, I think that would be our final question.
Yeah, I'm happy.
That's
for-
okay with you. Thank you, everyone. Put your hands together there for Professor Mario. So thank you, everyone. We will now move over to the concurrent sessions.